Hi, Eugenio. Can you hear me?
Yes, Wilson, can you hear me?
Yeah, sure. Yeah, I
Okay.
can.
Let's start then, right? I think we passed already 1 minute to after 2 p.m. Brazilian time.
Okay.
So let's see if people from the already the Ant 662 is already here. I see some names already. Uh, André is there.
You're looking at
So,
YouTube, right?
right, I'm looking at YouTube here. Socorro is here, Lohana. Uh, Peter is also here. Let me see if anything else. Tatiana is also here. Mateus, Luana. So we normally wait a few minutes because we have people from different countries. So
Uh-huh.
I wait sometimes 4 or 5 minutes before starting the talks. So,
Mm-hmm.
but let's start with saying that it's a very big pleasure to us here having you again
Oh,
at
I'm
this
back.
virtually in at the UFV, system. So, but I hope that in the near future you can come and visit us in face to face again. So, but yes, for I think for this moment on, you can start formally the talk. It's a big pleasure to us to have Dr. Wilson Valbon, here at the course Insect Physiology and 662 from the Federal University of Viçosa, in Viçosa, Minas Gerais, Brazil. Wilson Valbon, Dr. Wilson Valbon is a agriculture engineer. He got his bachelor title at the Federal University of Espírito Santo, Brazil. And he is a master and PhD. He did a master and PhD here at OFV at entomology program. We are so glad that you are now pursuing your postdoctoral experience at the Duke University in the USA. And Dr. Valbon is going to talk to us here about the neural basis and related behavior associated with the chemosensory chemoperception in insects. So, Wilson, it's a big pleasure again having you here with us. This talk is you are working here as a invited professor. So for this course, we are getting students here from Brazil, from Colombia, Nigeria, South Africa, and other parts of the globe. We are so glad to have you here. So as always the stage is yours, my friend.
Oh,
Thank you.
thank you very much, Eugenio, for the introduction. It's such a pleasure to be here today. I'm back. Unfortunately, not in person. I would like to be there in Brazil now at UFV, but not possible yet. Okay. It's such a pleasure being here today to talk to you guys. So I can see some names here that I overlapped there a couple years ago at UFV. So it's very nice see you guys again. So Eugenio, can I share my screen now? Can I
Yeah.
start? For sure.
Before you start, let me explain for the students here. They can stop you at any time, right? To ask a question, and then I will read the question to you here. Is that
Mm-hmm.
okay?
Yeah, not a problem at all.
So then the people can ask questions in English or in Portuguese, doesn't matter. We are having here big help, Wilson, from the Insectum, our graduate students group that is developing studies in entomology from our graduate program. Thank you guys for helping us here to
Thank you.
set everything up and to invite Dr. Valbon and to prepare all the things for this talk. And whenever you guys want to ask a question, please can stop, put the question here in Portuguese or in English, and I will stop Wilson and read your question to him. Okay, Wilson, thank you again.
Yes, no
You
problem.
can
Yes.
you can share your computer screen now.
Yes, let's get started here. I think I can share now. So can you guys see my presentation? Not
Not
yet.
yet.
So let's go back here. Let's share again. So share screen.
Now we are seeing you is.
Okay, there is a delay.
But it's not now again. It's off.
Oh, how
Try
about
again.
now?
Now is it yes. It's showing now.
Okay, okay.
Yes. This one exactly.
Okay.
Yes.
So can I get started?
Yes, please.
Oh yes. So this presentation should be last week, but Eugene changed for this. Okay, for this week, and today is a Friday, 13th, right? So let's see how this presentation gonna end. So Eugene made an introduction about me. So I'm located now at Duke University. Duke is a small and beautiful university here in the center of North Carolina. The city is called Durham. I think it is my. Hope you guys can see my cursor here. So it's a beautiful university here. So it's a Gothic-ish university. It seems that I'm inside the Harry Potter movies, even though I don't like Harry Potter movies, but look like I'm inside the movies. So it's very beautiful. And here's a picture of Amanda and I last winter. Yeah, we have kind of a little bit of snow here, not that much. It's very good, very nice environment. And you guys are very welcome to come here anytime. Okay, but the question is, how did I end up here in this place, Duke University? So everything started a couple years ago. So specifically started at 2015. So when I joined the graduate program as a grad student there, as a master student at UFV entomology program, here's a picture of my first party there. We have this like entomo party. It's like a welcome welcome party for the new grad student. The guys here with this blue face are the new student that time, and I'm here, right here. Professor Eugene is here, kind of different now. I have Professor Eliseo here behind. You have some face here. Ju is here. Um, we overlap there. So yes, then I started my master there, 2015. Then 2016, I started my PhD, and I developed half of my PhD project over there. Then in the end of 2018, I applied for a scholarship to develop part of my research abroad. I went to Michigan State University. So this is my my last picture in UFV, just to prove, guys, that I was there. So was my farewell party there. It was very nice day. So we get together there. So then this is my first self at MSU. As I mentioned, I stayed for like 1 year in Michigan State University because I was doing my sandwich doctorate from CAPES. There at Michigan State University, I had the opportunity to meet many researchers, good professors, and there I was located at Kedong Lab. Kedong was a professor over there. So then when I was there, she got retired from there, and she got this new position at Duke University. So then I finished my PhD. I returned to Brazil, finished my PhD. Then Kedong invited me to be a postdoc at her new lab at Duke University. That was how I ended at Duke University. Okay, so but life is not the only laboratory, is not the only science, not the only university. Look here, science is also life is also have fun. So this is a picture of me last summer. I was in Florida. I was enjoying for my first time this turkey leg here. Tastes very good, by the way. And as you guys can see here, I mean, I also enjoy a beer as well, because who knows me knows that I am a beer guy. But also there are some people that like to taste, to smell wine. Why not, right? But I still prefer beer. So And you have this fantastic ability of taste and smell things. Not only us human beings, but also other mammals. As you can see here, this squirrel is smelling this beautiful flower here. This picture is very famous around the world. And here you have this cute girl here tasting ice cream. Okay, so but how is this possible? You know that we can smell, you can taste, you can sense things, uh, and other mammals as well. So, but how about insects? Do insects can smell, can detect taste, something like that? So, this is the topic of my talk today here for this class. So, so yes, insect has the chemosensory system, and they have a very specialized sensory system that can detect taste and smell. And we call this gustatory and olfactory system. Okay, here we have this beautiful moth here. Very likely this is a male. This fantastic antenna here. This very likely is a male, and the male use their its antenna to detect the female. How? To smell the pheromone. Okay. Here we have a butterfly. This butterfly is suck up here nectar from this flower, but very likely before start to suck up, this butterfly tasted this flower with their legs. How is possible? Yes, butterfly use like legs to taste things, to try before eat. Okay, I have here also flies. This is Drosophila melanogassa flies. These ordinary flies we call fruit flies or just Drosophila is a model organism to study many things, and of course is a model organism to study chemosensory system. Okay, here we have also in this last picture here a beautiful female mosquito trying to bite this skin here. This female here is tasting. This is the labium to taste the skin to see if it's good. If it's good, she will bite. Yes, she because only female bite us. Okay, no male. So, and I'm very glad to talk about more specifically about mosquito today because there are many studies out there that they use these two organisms here, uh, flies or mosquito, as a model to understand the chemosensor system in insects. Okay, but this guy he is my beloved organism, my beloved tool to understand olfaction and the gustatory system. Okay, so then let's talk about a little bit uh, mosquito here. Then they are very well specialized in find host, and we are the host, right? But how? Look here. I record this movie here yesterday, and you guys can see here. I put my hand close to this net, close this cage. This cage have around 1,000 mosquito, and you have male and female here. And you guys can see that there are many female here trying to bite my hand. Look, they are crazy. They are very, 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 very eager, anxious to try to get blood, and they are very optimistic that they'll get. So this is the same video, but from another angle. Here, you guys can see here the mosquito are piercing the net. We can see the mouth part here crossing the net. Okay, but how do mosquito do that? How they do that? Uh, so to answer that question, we need to see the big picture, okay? Because chemosensory system is critical for the biological success of many species, okay? The organism needs to detect the environmental stimuli around. So, mosquito and other insects, they use the like antenna or maxillary bulbs to detect things. So here we have a picture of the mosquito head here, and they detect the information through the appendage here. They also this information is processed in the brain, in the brain level here. This is the central nervous system of insects. Okay, then you have the behavioral output. So the mosquito, the insect's gonna take the decision. Okay, I'll go there because it's a host. I wanna What I want to bite, I want a blood for my children. So, or you run away because maybe there is a repellent there. So then, this is not the topic of my presentation today, this brain level here, because this is this is nervous system topic, but I'm talking about today chemosensory system. Then I'm gonna focus on antennal detection, this peripheral system here, and also a little bit of the behavioral output. Okay, I will combine these two here during my presentation. So as I mentioned, they use the peripheral nervous system to detect things. And this information is going to be processed in the nervous system and then behavior. So here, this picture is very nice because here we can see which part of the mosquito head is detecting things. So here you have the antenna. Through the antenna, they can detect heat. They can detect also body odor. We have smell. Each of us has a specific smell in our skin that they can detect very well. And also they can detect things by eyes, by visual stimuli. So, and also CO2. I will talk about CO2 later in this presentation. Okay, carbon dioxide. They can detect carbon dioxide through their maxillary pulps here. Okay, here's just a zoom in of the tip of the antenna where they can detect the heat. I will talk about later uh, on heat detection as well. Okay, so let's move on here. Okay, but how mosquito detect such things? So in the antennae, they have like odorant receptor. Uh, they have ionotropic receptor they use to smell things. Okay, and in the maxillary palps, they have gustatory receptor to taste things, and they can smell through that by of odorant receptors and also ionotropic receptors. Okay, so for odorant receptor, we use OR to when I say OR is about odorant receptor, and IR is for ionotropic receptor, and GR is for gustatory receptor. Okay, so this receptor, I have a big picture here of these receptors. They are ligand-gated cation channels. Okay, they are expressed in the membrane of the neuron. Okay, the membrane of the neuron, the sensory neuron, has this receptor here. These receptors here. Then how things work here, right? So as I mentioned, uh, they use the antenna, and the antenna, if you zoom in the antenna of the mosquito, you can see several tiny hairs here. This is just one segment, this cartoon is just one segment of the antenna, and we have several sensilla. Uh, so each sensillum is a very small hair. This sensillum has like a membrane here, it has the pores, and the neuron, there are two neurons expressed, embedded inside this sensillum lymph here. You have A neuron in pink and you have B neuron in green over here. This is called sensory neurons or receptor neurons. In the dendrites, on the membrane on the dendrites membrane, the odorant receptor are expressed. Okay, the ORs or IRs are expressed in these dendrites in the membrane of these two neurons here. So, okay, so let's zoom in even more. Here you have the membrane of the neuron. Okay, here you have extracellular part, you have intracellular part, and here you have the cuticle, and the odorant must cross the cuticle and get in the lymph. So here in the lymph, you have other proteins. We have like OBPs that is responsible to transport these molecules, this odorant, to the receptor. But today I'm gonna focus only this interaction over here. Okay. So here the odorant is bind to this odorant receptor here, OR. And as you guys can see here, you have this part, or this protein here, and this second part here is called Orco. Orco is a short for odorant receptor co-receptor, okay? So it's Orco. Together they work. odorant receptor must be must be together with this co-receptor Orco here. Without Orco, will not work. And how it works? So once the odorant binds here, we have the influx of carrion inside the membrane, inside the cell. From outside to inside, we have an influx, okay, of carrion. Can be sodium, can be potassium, can be calcium, okay? So then this depolarization of the internal part of the cell is going to lead to action potential of that neuron, and this action potential is going to generate more action potential, and this information is gonna be sent to the brain. Okay, then the insect will make the decision. Okay, that's a very good smell. This is a human being. I'm going there to bite. Okay, so this is the how things work at the peripheral nervous system for odorant receptors. Okay, so then people are crazy because they are trying to understand much more about mosquito, and they realize they found out that there are more than 100 OR genes in Aedes aegypti mosquito. Okay, is the major organism that I'm going to talk about today, and however, odorant receptor co-receptor Orco is just one single gene. Okay, just one single gene, and they also always is connected with odorant receptor OR, and Orco is important. So keep that in mind. So then, how to measure the response of an insect to odorant, vapor compound? So here, I have a picture. I have this schematic figure here. I have electroantennogram. It's a very old technique. It's from 1950s. They figure out that they can record the response of antenna to odorant, to compounds. Basically, we can place the insect needs to be alive here. We can place the whole insect on the head. We can remove the head and place under a microscope, and we can use the antenna. We need to place an electrode on the in the tip of the antenna, and another electrode need to be connected to the eye. Then the circuit is going to be closed. Then when you puff a vapor here with a compound of just air, clean air, we can see the response here. There is this deflection in the response, electro response. What this software he is measuring? Is measure the electrical signal of that antenna. How? So as I mentioned before, there is an influx of calcium, of carrion inside the cells. Then everything outside the cells is gonna become more negative because positive things is going inside the cell. Then outside, this is a extracellular record is going to be more negative. Then if it's more negative, you can see this deflection here in the signal. Okay, so here we have the control when you apply just a vapor, clean air, no signal at all. Then when you apply the compound here, we have this deflection of the signal. Then you can measure the size of this deflection here and show and confirm, okay, that insect is smelling, is detecting that compound. And here, this bar here represents the application, the moment of the application. It's just half seconds, guys. It's very short. The time is very short, and even though they can detect, okay? So this is a very powerful technique that many people are using around the world, and as well in Brazil. Just to keep in mind that this presentation is about insects, not only mosquito. Amanda, my wife, had the opportunity to work with this technique as well in Brazil, and she got luck because her insect is very big. She worked with sugarcane borer, Diatria saccharalis, and she worked with the interaction among insects, among plants, and pathogens, because there are some fungi that can cause disease in sugarcane. Then she worked with the interaction, and she she exposed the antenna of the female moth to the compound that sugarcane produces. So then she got lucky because the insect is big. So because then she could remove the whole antenna and place it there in the EAG to see the response of the antenna. Okay, the same technique, different insect, very different insect. So If you go deeper, we can also record the response of a single sensillum, a single neuron. Okay. I showed you guys this image before. In the surface of the antenna, there are several sensilla, and we can place a very fine electrode inside the one single sensilla. Yes, it's very hard. We need to keep the insect alive under the microscope. And we can focus on one specific segment of the antenna, and you can zoom in and you can put this very sharpened, very fine electrode inside the sensillum. Okay, this electrode is going to make contact with the surface of those two neurons, neuron A and neuron B. Neuron A in pink and neuron B in green. Okay, then once the electrode is making contact, we apply the compound to see if you can see the response. Okay, so but here is slightly different from EAD. So here we are measuring the action potential of those two neurons, not the response of the whole antenna. Okay, in pink here you have the spikes. Each spike here, each line here represents one action potential. Okay, so then
Wilson, let me interrupt you because
sure.
Luana, one of our students here, is asking if if one of these if this insect lost one of these sensillas, it's gonna be restored, or how it's gonna be, how the insect deal with these losses.
Okay, good question. If they lose the sensilo not to be generated again, but the neuron that express in those the other one that express in those neurons is going to be expressing another sensillum. Okay, we have overlap of receptors. They not going to be a major problem if they lose one single sensilo, but the sensilo can't be generated again. Okay.
Thank you, Wilson. Thank you, Luana, for the question.
Yes,
Okay.
good question. Okay. As I mentioned, we have like more than 100 gene or odorant receptor gene that are expressed in mosquito, and you have several more than 100 sensillum. Okay, guys, many sensillum. They have a bunch of sensillum. Then, if you remove just one, not be a problem at all. But if you remove the antenna, yes, the whole antenna, yes, they can't smell. Uh, very well anymore. So again, go back. Let's go back here to this trace here, this single sensilla record trace. So we call this SSR. Then you have the trace here. In pink are the response of neuron A, and the green is the response of neuron B. Again, this is action potential of the neurons. The neurons are firing. It's just the control. We apply just a clean air, and we can see the neuron firing there because neuron there is always action potential happen there. Okay, and you can tell apart the difference between these two neurons by the size of the spikes. These large spikes are neuron A, and the smaller ones are neuron B. Okay, sorry. Then, okay, here I just applied a clean air, nothing, right? Then when I apply a compound, we clearly can see increase of the frequency of spikes here, especially these uh, pink spikes here, which means the neuron A is being activated by the compound I applied here. Again, this is just a half second of application. Then we can see here the response. Then you have the frequency of the spikes before and after the application. Then you can calculate the response of that specific neuron. Okay? So that's it. We have two major techniques: EAD, whole antenna, and you have SSER, single sensilla record, to measure a specific sensillum. Okay, guys. I forgot to mention that the sensilla is very hard to tell apart because they are very tiny, but they have different shapes. Different size. Then you can tell apart. Okay, that one is a specific type of sensilla. I will talk about this later. So then, and how about the behavior? Okay, now you guys know that how you can detect the response, the electrical response from antenna. And how about the behavior? How to measure the behavior? So in our laboratory here at Duke and before at MSU, we used this methodology before called arming cage, but we like improved this methodology. We call now handing cage. In this
Wilson,
methodology, okay.
Wilson, let me interrupt you again. Luis Salinas is asking here, good afternoon. Male and females of a single species can have different OBPs, OGPs, or Rs?
Yes, I mean, okay. Is a good question because there is a huge difference between males and females of ability to detect. I'm talking about mosquito now. Males also gonna have some odorant receptor, but it's gonna be different. And I mean, it's gonna be the same, but the number is gonna be different. The same gene, but in female mosquito have more than 100 that's being expressed. In male, it's gonna be much less because they don't need to smell human. Then it does make sense express these proteins over there, but it's the same in both male and female inside the same species.
Let
Okay.
me just to ask if Salinas, if you want that we translate it to Portuguese, let us know. I forgot to ask that for Luana as well, but Luana, if you want that we translate it, just let me know, okay? Just
Yeah,
pause.
yeah, yeah. I'm much better in Portuguese than English. Yeah, then we can
Are
speak.
you sure?
Yeah, yeah. For my Portuguese is much better than my English, for sure. So, can I go back?
Yeah, please go
Okay,
ahead.
thank you. Then yes, then you have this methodology handy cage here. Basically, we place our hand inside a cage with several mosquito. But how? How to avoid the mosquito bites? So we have this glove here. This glove is modified. We need to wear the researcher must wear this glove. This glove has a hole on top of the glove. Then on top of that hole, you can put a stack of magnet frames. Then here we have in between these magnet frames here, we have two nets. Okay, we place this first bottle net here. We apply a compound here, we apply a solvent or a repellent here. Then half inch above this first net, we have a second net. This second net is untreated net. We don't apply anything here. This is just to avoid two things. First thing, avoid the mosquito make contact with the compound here because we want to make sure that this is spatial repellency, not contact repellency. And also to avoid the mosquito bite us because guys, there are 40 very hungry females inside this cage. If they start to bite us, it's crazy. So then we need to put this first net here to avoid the mosquito bite. Okay, and again, this bottom net here is treated with insecticide, repellent, or essential oil, but it's not making contact with our skin. Okay, there is space enough here to not make contact. Then we place our hand inside a cage. We have a camera on top of this cage, and the camera will record the behavior of the mosquito. We have something like that. Eugene is very aware about this methodology here. Then we can count the number of mosquito that is trying to bite us here. Here we have several mosquito. We put around 40 mosquito. I think almost all of them are here trying to bite the hand here, very like this is my hand, and you record this for 4 minutes. Just to provide more information about this methodology, we evaluate the number of lanes in 2 moments. We have the first trial. In the first moment, we offer our hand with the bottleneck treated with solvent only. We use most of the time we use acetone here. Acetone is a very good solvent. Wait the acetone evaporate, then we place the acetone in the glove and place our hand inside the cage. Then we count the number of lanes during 4 minutes. Okay, then you wait. Then this one single cage. We do this with 10 cages. Then we wait 1.5 hours. When they have this interval, it's time to mosquito recover from the first exposure. Then now with the same bat of insect with the same cage, we will expose again, but now the bottom net is treated with the compound. Okay, then we need to count again the number of lanes. Then you can calculate the the behavior, the output. We calculate the total number of lens in the second in comparison with the first one. Then you have the repellency index that I'm going to show you later here. Then you have baseline. Have baseline should be close to 0 or around 10% of attraction, 10% of repellency, okay, because it's just compound, acetone on the first trial and acetone on the second trial again. When you try the second trial, the repellent, then you have a reduction of this repellence, of this length, okay? Then you can calculate the repellence index. So then in our laboratory here, we started this repellency project 5 years ago or 6 years ago. I wasn't here. At that time, Kerr was in Michigan and I was in Brazil. I was not thinking come to US yet, and she started this project over there, and she used this repellency spatial repellence assay to, to measure the repellency of Pyrithione extract. So again, here is just a picture to see again the how is the setup, and here you have the response, the output. Okay, in the x-axis here we have the treatment, we have like control, we have a Pyrithione dilution time 10 minus 3 and 10 minus 2, and here in the y-axis we have the repellency index in percentage, okay, the percentage of repellence. The black dot is each case with 40 mosquito each, okay. Then you see for control we have a little bit of repellency, but it's like baseline. It's nothing. It's a normal background for the mosquitoes. Then we apply when the bottleneck is treated with pyretron. We have this part at 10 minus 3. We have it around 40% of repellence, and 10 minus 2 around 70% of repellence. Then we can see that the repellence is concentration is diluted dependent. Okay, if we increase it here, we increase the repellence. Okay, then, okay, this was the first step of this project back a couple years ago. Then we asked ourselves, why?
Wills, before you move on,
Uh-huh.
David is asking, and I would like to come back to the topic, earlier topic. He's asking, then it's possible then in the same sensilla, when the mosquito loses the hair, could there exist another sensory receptor like mechanosensory?
In the sensillum?
Yes, in the
No,
sensilla. He paused in the sensilla, but I think he
no,
meant.
the mechanoreceptor are in another place in the antenna. It's more close to the base of the antenna, okay? In that sensilo, there are only odorant receptor or ionotropic receptor, specifically for smell things. Those ones that I'm showing here.
So just to make clear, you are saying that there's a sensilla that works like for smelling doesn't have any impact in the mechano perception of the
No,
environment.
no. In this case here, no.
Okay, thank you. I think
For
it's clear
mosquito.
for
Okay,
very.
okay, okay.
Thank you.
Thank you. So yes, as I mentioned before, you could see that the pyretron extract is causing repellency, but which is pyretron? So pyretron is is a mix of several compounds. You have this pyretron extract here. This extract is collected from this beautiful plant here, chrysanthemum flowers. People in Asia many years ago, they dried these flowers and they make this extract, this crude extract, and they realized that this is a insecticide compound. This pyretron extract kill insects in general, different types of insects, right? And also they realized that pyretron extract also repellent. So, and after that, people start to use pyretron extract as a repellent. And as you guys can see here, you have this mosquito coil here, then you can burn this, and this will release the vapor. Here you have this smoke here that is represent the vapor from this coil. And the mosquito will run away because they don't like it. The the pyretron extract came from this coil. But how? Even though this compound, this extract has been used for many years from the from since people that start colonize, people start living in Asia, they didn't know. People didn't know yet how this pyretron works as a repellent. we know very well how pyrethrine works as an insecticide, but as a repellent, no. Just to make clear, guys, pyrethrine is the inside the pyrethrine extract there is pyrethrines, okay? And pyrethrines is the structure of pyrethrines was used to make this very famous insecticide that's called pyrethroids. That also are repellent as well. People use pyrethroids as a repellent as well. But until now, people didn't know how pyrethrin and also pyrethroid work as a repellent. Okay, so then we worked for a couple years try to answer this specific question here. Okay, is the repellency, the Pareto repellency odorant receptor mediated, is Orco mediated? Are the mosquito smelling pyrethrine extract? So if yes, which odorant receptor neuron is being activated by pyrethrine? So, okay, if yes, which OR is expressed inside in that neuron? Then we worked hard here for at least I worked for 2 years, and other people worked for 5 years or 2 more years to try to answer this question here. So now, before I go to the results, I need to make a short introduction. I will introduce this mutant line, mosquito line. We call this Orco. I asked before you guys to memorize this name here, Orco, because Orco is a co-receptor, right? It's odorant receptor co-receptor. It's always combined, always together with ORs, and it's just one single gene. So if it's just one single gene among the 14,000 that are present in mosquito, we can delete it. Then people, couple years ago, they deleted the Orco gene. Then we have a mosquito that does have a mosquito line that does not express Orco protein anymore. Then what happened with this line? Very likely this line will not smell anything more related to anything that binds to odorant receptors. Okay? So then we tried this line in our lab to see if the Pareto repellency is OR mediated. We use again hand cage, we use the Orco mutant line, and here we can see the results. We have the repellency, we have the treatment here control, and we have the Pareto 10-2 dilution. For control, no difference between two lines, the wild type and Orco, wild type in green, and the Orco in white. So here we can see that the 10-2 dilution at least around 80% of repellency in wild type, and in mutant mosquito, this was dropped to around 20%. There is a very clear, sharp reduction of the repellency here. Then, yes, the Pareto repellency is OR mediated. Something inside the Pareto extract is target odorant receptors. So then we went back to EAD to see the response of.
Wills, let me interrupt you again.
Sure.
There are two questions here. Ezra, Abba, and Tatiana Bernal both are asking questions regarding the paratrum.
Mhm.
So first, Ezra is asking, is it synthetic pyrethroid used in the experiment? I think we can. I already answer his question. I said I know that pyrethrum is a natural compound.
Mhm.
Extract from Pyrithione, from
Chrysanthemums.
chrysanthemums plants. But Tatiana is asking, how safe is Pyrithione extract for humans, and how was its use evaluated?
Oh well, yes. Among the insecticides out there, this group of insecticides called pyrethroids, they are the safest one because they have low toxicity to mammals. Okay, and does not this molecule does not persist for a long time in the environment when you compare to DDT, for example. And they are no it's not very toxic to bees when you compare to other insecticide. It's kind of safer. And people made before you develop any insecticide, we need to try in several organisms, and they are not toxic for mammals. The receptor that pyrethroid targets is different in mammals and insects. Then in general is safer. Okay. The other question was, Eugenio. Yes, pyrethrum is a natural, is extracted from plant. Okay, and pyrethrum is gonna be even safer than pyrethroids because pyrethroids is they modify our pyrethroid to become more potent as an insecticide. Okay, then yes, you have these two: the natural one, pyrethrum extract, and the synthetic one, pyrethroids.
I think you might show some pyrethroid, synthetic pyrethroid results during
No,
the.
no, no. Actually, I won't. I won't because I need to go to other topics. I'm not
Okay.
focused only on odorant receptors. Okay, but we can discuss more at the end of this presentation. Okay,
Yeah.
because we
Yeah.
also tried pyrethroids. It's kind of similar.
I
Uh
think that Ezra interests because he's working a lot with mosquitoes over there
huh.
in his home country. So then I think he might place some other questions. Thank you, Wilson. Sorry for
Okay.
this interruption.
No, no
Interrupt.
problem. You are here for that. So
Okay. Thank
go back to here to EAD. Now we applied pyrethrum here for we delivered the pyrethrum vapor to wild type mosquito and Orco mosquito. Here you can see I showed this before. Pyrethrum, when you apply pyrethrum, there is a deflection here of the signal. And when you apply pyrethrum to Orco mosquito, nothing, which means they are not smelling. And very likely it's only odorant receptor mediated. Because if you pyrethrum target, for example, IRs, ionotropic receptor, it would be possible to see a small peak here at least, but nothing here. Then yes, then we kind of confirmed that the Pareto activate odorant receptor neurons. But the question is now, which odorant receptor neuron is activated by Pareto? So then we will use it single sensilla record, as I mentioned before, have many sensilla expressed in the antenna, and you can tell apart the sensilla by the shape. These have a short, blunt tipped one, so it's not like a sharpened sensilla. And we apply a panel of several compounds also to see the function of the sensilla. Then you can tell apart which sensillum is being activated by a specific compound. Okay, have it by structure and also by function. So then we could see that SBT1 sensillum, short blunt tip sensillum, is activated by Pyrithione here. As you guys can see here we have an increasement of the spike frequency for Pyrithione here in this A neuron and nothing for B neuron. Okay, we found that at least one odorant receptor neuron is being activated by Pyrithione extract. Okay, but now what is the identity of that neuron, or sorry, of that odorant receptor that is expressing that neuron? Okay, we found the neuron. Then what is the identity? So this is a very hard. There's a very technique to find that. After a couple of years, we could find. I'm making here a long story short and show you guys that we identified the identity of that odorant receptor is odorant receptor OR31. OR31 is expressing that sensilla that in that neuron that is activated by pyrethrum. And just uh, to let you guys know, this odorant receptor here is conserved among the mosquito species. They are present in Culex and also Anopheles. They have orthologs in those two species of mosquito. Anopheles is the malaria mosquito. Culex is the another mosquito that also transmit disease, and Aedes aegypti is the yellow fever mosquito and dengue mosquito and Zika and the other disease. Okay, so yes, we found out, we discovered the identity of that neuron here, odorant receptor water one. Okay, bingo. What was now the next step of this project? Then you decide, okay, let's confirm if in vivo, if the behavior of the mosquito will change if you delete the OR31 gene, if you delete this receptor. And here, now we use this the CRISPR-Cas9 system to make the knockout of this specific gene among 14,000 genes present in the mosquito genome. We delete just one here, water1, and the mosquito could do everything perfectly. This deletion was very good, very well done. Then we developed this mosquito line that's mutated. However, when you use the pyretron to try the repellency in these mosquitoes, the repellency is reduced. Then you can see here in blue we have the wild type mosquito, and in white you have the mutant mosquito, water1 mosquito. For control, no difference. The background of the mosquitoes are perfect. They are not sticky. They are sticky. So, and yes, the repellency was reduced in water1 mosquito. Okay, then we confirm that water1 is a very important odorant receptor. So we asked ourselves, so is water1 activated by other repellents? We talk about here about pyretron, but how about other natural repellents that's available out there? So yes, we got lucky here because besides pyretron, water1, the same silo here that express water1 neuron is also activated by citronellol and geranyl acetate. Citronellol, I think, is among the most famous natural repellents present out there. Also insecticide, I think so, but it's very famous as a repellent. You can go to the market here, you can go to Walmart and buy a citronella grass or citronella plants. This plant here smell citronellol. That's the major compound over there. We also can buy candles. I have a candle here in my backyard that when you use the candle will release this citronellol smell and the mosquito will run away. And they also have geranyl acetate, is another natural compound. So yes, OR31 is activated by these two compounds as well. And also the repellency. This is very interesting, guys, because the repellency, again here the control, again here the pyretron was reduced in OR31 mosquito in these mutant guys, and it also was reduced for citronellol and geranyl acetate. Okay. Yes, OR31 is the first conserved mosquito odorant receptor that has been demonstrated to mediate repellency. And we were the first research group that deleted a specific OR gene to make this, to change the behavior of mosquito to reduce the repellency. We'll talk about repellency, no attraction here, okay? Then you use this, we combine several approaches, several tools to confirm that the pyretron repellency is odorant receptor mediated, and we found out the specific OR that's activated by pyretron. And if you guys want to read about these finds, you can go to this paper here that we published last year at Nature Communication. We have a lot of things more there, more interesting things that is not the topic of this presentation today. It's just odorant receptor part that I showed you. Besides Eugene and I, that from Brazil, from UFV, we also have another guy here, very talented guy, Felipe Andreazza. I think some of you guys know this guy here, Felipe. Felipe is my friend, is my laboratory colleague. We work together here. Very nice guy.
Will,
Hey,
I I have another question here
sure.
before we move on. Vanessa Herrera is asking here if there is any risk for resistance to pyrethroids in mosquitoes, and if there is such risk, how would it be, how the chemosensory would be affected, how the functions of the chemosensory system would be affected for this?
Okay, it's a great question. Great question. And the easy answer for you is asking you to read the paper. But I'll talk about here. Yes, when the mosquito are resistant to pyrethroids or pyretron, the mortality, the repellency can be reduced because of this mutation on sodium channel. That's the major target of pyretron as a insecticide. Okay. However, this mutation on sodium channel won't change the response of the odorant receptor. The repellency that is mediated by infection of odorant from odorant through odorant receptor will not change, won't change in resistant mosquito. Okay, in pyrethroid resistant mosquito. Okay.
So
Uh,
You are saying that there are some pyrethroid resistant mosquitoes
Yes, there are. Yeah, many, many
that
lines.
they have chemo perception affected by such
Mm,
mutations. Is
No,
that correct?
no. The chemosensor ability is not reduced by this mutation. Okay.
But okay,
Okay, but as you can see here in the title of this paper, there is a dual target molecular mechanism. I just showed you one of these dual. I just showed you the odorant, the olfaction mediate. Okay, this second one is by targeting sodium channel. It's another topic. It's another. Yeah, I need another class. And I need another day to talk about that. But the second target here, sodium channel, yes. Then the mutation, the resistant mosquito is gonna behave different. Okay.
thank you, Wilson. I think Vanessa, if you want to translate to Portuguese, I can help. For Spanish, my portuñol is muy mal hablado.
Oh my goodness. I
I
don't speak Spanish. Sorry. I this is my major goal for the next years, for the future.
Thank you, Vanessa,
learn
for the nice question. Thank you,
learn
Wilson.
Spanish. Okay, okay. So yes, and here I finished this first part about odorant receptor. Yes, we can use olfaction to show how repellent works. Okay, but let's go to another topic now, another different way. So again, as I mentioned before, mosquito can detect heat, can detect the, carbon dioxide, and the uh, body odor. And the body odor is the same way that they detect pyrethrum. Okay, is by targeting odorant receptor. Then I'll not cover this anymore. Okay, but now I'm gonna focus on heat and the carbon dioxide.
Before you
So
move on, Luis,
sure.
before we move on, I think Vanessa is placing another very interesting question that I could leave it for the end of the talk, but it's a nice question. So
Mhm.
she's asking, what do you think about genetic modification in mosquitoes for population control? Is it safe? Taking into account the resistance problem, would this be a good alternative for you?
Oh, great question. Great question. These are more is a how to apply these techniques in the field. Yes. So I would say 99% of the research with this generic approach is doing in the lab just to show how things work, to show the function of a receptor, to show how an insecticide works, how a repellent works. Uh,
Will
in,
just interrupt you. I think she's asking about the in the general the mosquito control by releasing mosquito that genetically modified.
Yes, I'll get there. I'll get there. And
Yeah,
also
okay.
is using this approach to develop mutant mosquito and try to release these mosquitoes on the field. Like you have gene drive that can rescue the susceptibility to insecticide. Then the population in the field that are resistant is you will cross with this line, and the offspring is gonna become susceptible as well to insecticide. Oh yes, it's kind of possible, but I would say there are a lot of study that need to be done more, and it is very it can be useful for a small area, and also like islands that you can control everything. Because you think, okay, you are trying to develop a mosquito in the laboratory that is mutated. Okay, the overall fitness of this species, these guys in the lab, it's okay. But how about in the field? Will this mutant insect be able to mate with the guys outside? How about the competition with the other guys in the field that they are established already? Plus, when you for example, you release a population of mosquito, a mutant mosquito in the field, for example, mosquito that has Wolbachia, for example, not a mutated, but they can introduce Wolbachia. Will this mosquito be tolerant to insecticide? Because people will apply insecticide to kill mosquito, and these guys that you are releasing, maybe they are more susceptible than the guys in the field. Then you have this overall. I mean, it's we need a lot of study to combine things. And also, when you release a mutated species, a genetically modified organism in the environment, things can change. Okay, then we need a lot of study more to see these ecological effects in the population, also in the community, in the ecosystem. Okay, your question is very good, but it's still working in the lab. Outside is not very feasible yet. Okay, Eugenio, are you there?
Yes, yes. Okay, thank you for answering that, Wilson. I think yes. Let's see if she has
Yeah,
any
if she
compliment.
has.
We ask the at the end.
Yes, yes, because I answer in an overall way, a general way. Okay.
Okay,
So,
thank you.
no problem. So here now let's talk about heat and carbon dioxide. Okay, how these guys detect this. So, guys, just to remind you, everything that I'm talking here is also uh, true for other insects. Okay, flies also can detect. Uh, Others can detect CO2 and can detect heat. And most of these studies start with flies. Then people use the flies as a model. Now we are applying this to this for mosquito. That's a very important model as well. So then I was working in the lab last year, and I was read some paper, and I found this methodology here to measure the attraction to heat. It's a very feasible approach. Here, basically, we have like a peltier element here. This peltier is like this square here. This square can be plugged in a power supply. Then you can change the voltage here. You can control the temperature here. Okay, you can, if you if you provide more voltage here, we increase the temperature here. Okay, then you can you can control the temperature of this peltier element here, and you can place this peltier against a wall here. Here I made uh, this wall area here. You have this white wall in the back and two here in the lateral and in the sides here. And I place the peltier here. Again, I place here the peltier. I put this peltier, the size of this area here is around 7 to 6 centimeter. Sorry. So then I also I can apply CO2, carbon dioxide here. Okay. Then I can place a cage with some females against this peltier here. And you can see that the mosquito is going to be attracted to the heat area. Look here, this move. You can see that the number of mosquito will increase here rapidly. So, but why? If I'm looking for attraction to heat, why I need to have CO2? I will explain later because without CO2, they don't do anything. So I need to combine CO2 plus heat here to have this attraction. Look here, they go there, they try to bite there. These guys are very optimistic. They are think that there are some host, there is someone there that they will get blood for their offspring. Okay, but it's not that true. They are not so smart. So there is no one there, just heat and CO2. So with that, I tried like 4 different temperatures. You have the temperatures in Celsius, not Fahrenheit, and here in y-axis you have the normalized number of lanes. I could count the number of lanes. I record the mosquito behavior and I counted the number of lanes. And here you have for the wild type mosquito. So here you can see that for 36, why I start 36, guys? Because 36 is optimal temperature for mosquito because it's very close to our temperature. The temperature of our body is around 36. Then yes, they like this temperature here, as you guys here can see. I tried many cage here, many black dots here, and around 200 lengths. Then I start to increase the temperature, and 45, 46, 56, and 66. And you have this very clear reduction of this attraction here. Why? Because this temperature is not good for them, it's kind of nauseous, then they will avoid. Then you can see here that there is this temperature dependent response here. When you increase the temperature, the response is reduced. Okay, but how mosquito detect uh, uh, CO2, heat? So we don't have this question yet for AIDS Egypt. I'm trying to find this for AIDS Egypt, but we do have some cues that came from Anopheles gambiae, the malaria mosquito. They can detect heat as well, And this a very nice paper from Brandai University. This group of research, they found out that the ionotropic receptor 21A, now I'm talking about IR, okay, is responsible for detect heat in Anopheles mosquito. He's just a cartoon showing the antenna, the tip of the antenna. In the tip of the antenna now, it's not in, it's a different type of sensillum, okay? In the very end tip of the antenna, we have this coeloconic sensilla, and inside that sensilla, we have receptors. And here you can see the wild-type, these receptors expressed in the very end of these neurons, in the dendrites of those neurons. And here, in this axis here is the body. Let's see this central image here. These asterisks here are the cell body, and here the arrows are the receptor, or where the receptor are expressed. Okay, and here just the control. Here we have the mutant guys. They mutated. Yes, they found out the receptor, the IR21, and they made the knockout. They deleted this, and there is no more here the receptor over here. Okay, and here, they use a methodology similar to our methodology. Actually, it's the opposite. I use a methodology like similar to their methodology. So they have this cage with several mosquitoes. Have here Peltier in two temperatures, 26 and 37, and you have you need to provide CO2 here. So here, for example, for wild type, let's pay attention in this left here. Yeah, figured here. So here you have the environment CO2 and the two temperatures. Look, the mosquito can't tell apart. They are not motivated. They need CO2. CO2 is a trigger. Okay, CO2 is the trigger for mosquito to detect, and the CO2 that came from our mouth, our breath, they can detect. So then, after apply like 2 minutes of CO2, yes, look how many mosquito are here in this Peltier at 37 degrees Celsius. Slow, yes. We found something very consistent. Then when they use the mutant line, or R31, IR21, nothing there. And they use they created two lines just to confirm. Okay, they use two approaches to develop these two mutant lines, and for both, nothing. They can't detect more or reduce it a lot. So yes, so ionotropical receptor 21 is important for heat detection in Anopheles gambiae mosquito. How about Aedes aegypti, yellow fever mosquito? You don't know yet. So another hood of IRs is also to detect others, like odorant receptor. IR also can detect others. And like there, similar to Orco, we also have here IR8. Here should be Irco, because IR receptor, ionotropic receptor, co-receptor, but they call this ionotropic receptor 8A. This is the co-receptor, okay? I think, yes, in Aedes aegypti, now I'm talking about Aedes aegypti mosquito now. Aedes aegypti mosquito has like three different genes that can be co-receptor for IRs receptors, and one of them is this IR8A receptor. So, and people, again, this it's a very interesting paper. This paper came from Europe, I think so. They deleted, like the people made with Orco. They made the same. They use the same approach here to delete IR8A. And what happened? So then we also always we need to prove the behavioral response of the mosquito. They use like this uniport olfactometer, when they use a hand here and they provide CO2, and they measure the attraction here to this area, mosquito that were attracted to this hand. So here you have the percentage of female that were attracted in Y, and here we have the treatment. We have like wild type, we have IR heterozygous, we have Orco, 2 lines of Orco mutant mosquito, IR homozygous, half of the gene is deleted here we have half of the receptors and he is completed because it's homozygous, and also they combining IR and Orco. Okay, and each color here is one single individual, one single person. Okay, so here for wild type and for IR heterozygous and for 2 Orcos, mutant is the same. Okay, even though this line here Orco doesn't have the co-receptor for Orco, they still can find the host. How? Very likely because they have the uh, IRs that here is not present anymore because people deleted this IR8 here, this co-receptor, and yes, was reduced the attraction to this hand here. And the when they combine IR and Orco was reduced, but not different. Okay. So they also used EAG, as I mentioned before. They tested several compounds. They test like miscellaneous of compounds here, several different type of compounds in wild type mosquito, and they also tested a second group of compounds that are acidic volatiles. So here in pinkish here. So yes, the wild type responds very well for all of these compounds here. However, when they tried the IR mutant mosquito, they respond very well to these different compounds over here, these miscellaneous of compounds here. However, the response to um, acidic volatiles was reduced here. Look, in gray, was reduced when you compare with wild type. And among these compounds that they tried here, I would like to highlight this lactic acid. Lactic acid is a very common repellent that is present in our skin. This compound here is present in our sweat. Okay, we sweat a lot, and this sweat has a smell that mosquito can detect, and they can detect this lactic acid here. Okay, so very likely lactic acid, yeah, not very likely acid that binds to ionotropic receptor IR, and they just made now another behavior say here, another round of behavior say using this uniportal factometer again, but now instead of a hand, they applied CO2 plus acid lactic. And you can see here for wild type blank nothing, wild type only CO2 nothing. When you have wild type CO2 plus less lactic acid, yes, we have attraction, not that much because just one compound. Sorry, lactic acid alone can elicit attraction, but not that much. However, the most important thing here is that when they tried the IR mutant, this was reduced. Then they confirmed that lactic acid is activating IRs. Okay, so I talked a lot about CO2, carbon dioxide, but how mosquito detect this? So it's different now. It's not IR or other or OR. It's now GR.
Wilson,
So
just to
yes,
another question. Maria do Socorro was asking a few minutes ago if there is any perspective of extrapolating these studies to other insect vectors or pest groups. I
oh
mean,
yeah,
she was talking about the chemosensory systems and stuff like that. If there is anything already concrete, being conducted maybe by your lab team or by other people that you are aware.
yes. As I mentioned before, the first find about this receptor was were in flies. Then in flies, the Drosophila we like stole this from flies, and now you are using mosquito. Okay. Yes, there are study in moths as well. It's kind of the overall idea is similar, but it has some peculiarities that we can discuss more later. Okay.
Thank you, Wilson. Thank you. Thank you.
Mhm.
So we can talk a bit about that after the talk.
Yeah, because let me talk about GR now, gustatory receptor. Okay, as I mentioned before, gustatory receptor is present in the maxillary palps of mosquitoes, and here this group from the Rockefeller University, they made this line here that is lacking this GR3 here. Then let's go here to wild type mosquito. Then, for example, when they use the same approach as I mentioned before. We have this Peltier that have the temperature around 37, and we need CO2 to activate the mosquito. Yes, wild type, when we apply the heat and you have a puff of CO2, yes, we have the heat seeking here in percentage. We can see here that there is the mosquito go there to that area to try to bind, to try to get blood. Okay, only you have heat. Okay, when you remove one of that, for example, when you provide only heat or CO2, nothing. The mosquito won't go there. So why? Because this should be combined. It's a multimodal mechanism. Okay, they should be fused. Okay, combined. Alone is not able. So then, using this mutant line, GR3 line, the mosquito now is lacking this attraction to heat and carbon dioxide here. Okay, they are not smelling, they are not tasting CO2 anymore. Guys, it's also important to say that, for example, this gustatory receptor is also responsible for flies detect carbon dioxide, and also gustatory receptor is responsible for taste like bitter and sugar and other compounds. Okay, not only carbon dioxide, other things as well, especially compound that's present in food for flies. Okay, they can taste this. So to sum up here, the overall picture of my presentation today is to show you guys the superfamily of chemosensory receptor. We have ORs, odorant receptor, we have ionotropic receptor, we have gustatory receptor, and the function of these insects or odorant receptor ORs is for food odor and pheromone detection, and IR is for also for odorant detection, and especially acidic volatiles, sugar as well. Sorry, no sugar, heat as well here. And the GR is this gustatory receptor is to detect CO2, the carbon dioxide, in several type of mosquito. Okay, combined these two here is called olfactory sensory neurons, and here is the gustatory receptor neurons. Okay, that's all for today, guys. And just to finish, I just want to highlight again that insects are very well equipped to detect things in the environment. Okay, as I showed this before, I'm just showing again. And today I talk about some specific things that they have that make them able to detect the environment. So I want to finish with a big thank you for for this opportunity, Eugenio, and for Insectum as well to make this available in YouTube on the YouTube. So here's a picture of UFV. Every single day I cross it this grass here in UFV. This is close to the new biology department, I think so. Eugene Laboratory is very close to this area here. This was my everyday view going to the lab. Thank you guys so much.
Okay, thank you, Wilson, for your talk. Now we are open here for further questions that people can place here at the
Mhm.
YouTube, and we can, you know, go ahead with the questions and stuff like that. Okay,
Oh,
let's see.
now can I stop sharing now, or should I stay here with the PPT? my
It's
presentation.
up to you, I think. If you want to stop, I think there is no problem. Then we can share again if there is some need for sharing
Yeah,
stuff.
because I want to see people. I just see my
Okay,
presentation. I want to see people.
that's okay. That's more than okay. Let's see if there is any other question. So far, no question here. I think that I tried to cover all the questions
During the
that
presentation.
were showing up during the presentation, Wilson.
Hmm.
So, but let's open here for discussion. I think there are some topics, very interesting topic that show up, and people can question right now. Ok? Pessoal, vocês podem fazer perguntas em português, se preferirem também, a gente traduz aqui. Até no espanhol, se depender como for, dá para a gente ir no Spanglish e desenrolar aqui também, tá? Fique à vontade aí. Wilson, normally it take a while for people getting, the questions to prepare here to us. But I was a little bit, curious to see how the combination of how this heat perception together with the chemio perception could interfere with the let's say the pyrethroid or either any other insecticide perception. Are you guys invest some time trying to link these two teams to research topics?
No, yes, that's a good question. We kind of are doing that because I showed you guys with pyritron. Pyritron has a specific receptor, right? Then is affection. But however, in pyritron has another, it's a compound there, it's called pyritrin, and that's very similar to pyrethroid. And yes, pyrethroid also elicits repellency in mosquito, but we don't know yet how. And very likely, or can be possible, I don't know, not very likely, can be possible that pyrethroid is reduce the ability of this mosquito to detect heat, for example, or detecting CO2. That's one guess, for example. But we don't know yet. We know that pyrethroid targets sodium channel. Then there are some very sensitive sodium channel in mosquito. Then when you apply very low concentration, because pyrethroid, the high concentration is an insecticide. Pyrethroid, the low concentration is also a repellent. Then with very low concentration, we need very sensitive channels that should be activated, should be uh, 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 targeted by pyrethroids. But we don't know if that's activation of that specific activation is linked to this reduction to attraction to heat, for example. We don't
Okay.
know yet.
Thank you. I'm just smiling here because John Lopez, one of our graduate students here, say that Spanglish I went beyond the reasonable aspect. That he's joking, making joke with me because I'm not able to speak Portuñol. Can you imagine speaking Spanglish?
Spanglish, yeah, hard.
Thank Very hard. Okay, guys, questions. I'm expecting questions from people from the class as well. Otherwise, I will ask them questions. You guys need to decide. Either you place the question by yourself or I will place question to you. All right. Peter, any question, Peter? Andrea, let me see. So who else is there? Ezra. Ezra is already asking question. Uh, Socorro also did ask question here. So, Wilson, I think that we have a few minutes more if you want to talk anything that you planned but was afraid that the time was not sufficient. The stage is yours again, but as you might be aware, we have 1 hour and 40 minutes here. We're almost there. Thank
Yeah,
you very
no more question. I think people don't like. Like they didn't like my presentation.
No, I don't. I will not say that. Normally people leave the questions for the end, but I was, interrupting you all the time to make questions. I think that they did already a good. Your talk was nice. It was pretty clear what you're saying here. You make a very complex team, very understandable for most of the people here. I'm more than sure about that. And uh, they they can ask a question here any moment if they want. But if not, I would like to thank you very much for the talk, for the presentation, and for the motivation. It was a big pleasure, as not only because you work with our team here at the Viçosa, but as a professor of the graduate program of entomology. I'm very glad for having you here today, and I hope that we can do it every year now that we learn a lot with the pandemic situation, right? So
Yeah,
that we can do it more frequently, not only with you, but all for the people that are taking classes this year. They will be very welcome to share their knowledge, their interest in the future here as well in this course. So my plans and the plans of the graduate school is every day go more and more international.
hmm,
So we are going to have this possibility of doing these remote classes. So be prepared that we are going to ask you to be, serve as an invited professor here in this course again in the near future.
okay, okay. Thank you for the opportunity. It's a good train. It's a good train for.
Okay. Thank you. So Cole is saying here that you did an amazing work, Wilson. Thank you very
Oh,
much.
thank you, thank you. Yeah, guys, don't be afraid to make question. Okay, can I can talk in Portuguese. I can talk in Spanish, but maybe I can understand the question. But I will talk in Portuguese or in English. But
And some of you, I don't know, Wilson, but you might be coming to the Brazilian Congress of Entomology next August.
I don't know. I don't know yet because I need to extend my visa, my visa situation I'm deal with now. And I'd like to go because I'd like to go to Brazil. It's exactly 2 years that I came to US and I'm here. Then yes, I don't know yet. I don't know the answer yet. But we can discuss more
Okay, Luana
another
also
time.
did a question and she participated on the course here as well. Andrea, no question, Andrea. He's saying that there's a lot of moth experiment over there as well. So you are working. Oh, okay, you're working with the cotton, right? It's not. I thought it would be maize or soybeans,
I
but
see.
yeah, it's not so many.
Who are the person from Africa that works with mosquito there?
Ezra is from, I think, from Nigeria. Peter is from South Africa. And
Hmm.
David is from Colombia. The three that and Stefan was asking here to recording the presentation because he was having some trouble in the power supply in his home country. So he might come back to see the talk later on in the YouTube.
Okay,
Okay.
sure.
Guys from Insectum, thank you very much again for all the help, for the connection that you guys did here to place this talk here to facilitate my and Wilson's lives, and teaching us, educating us, how to manage these platforms and stuff like that. You
Yeah, it
guys
was my really first time.
You guys are doing an excellent job here at the Graduate School of Entomology. Congratulations. Again, as a professor of this program, I feel very, very happy to see how these young people are doing an excellent and amazing job educating us here, professors, to manage this stuff in a decent way. You think that you can do it, but once you are challenged to place a talk and to interact in a live, so you see that you know nothing. So without your help, it would be much more difficult to us here. Thank you
Yeah.
very much. Right? And
Yeah, there are
Wilson,
some. There is someone else that asked here: Is CO2 detection a function of olfactory or gustatory, or both? I answer here is a gustatory receptor in insects. As I know so far, is gustatory receptor.
okay, William is also asking, thank you, thanks for the presentation. John Lopez, many thanks for the Wilson lecture. You could tell me a little bit about the mechanism of action of the proteins that bind to the odors, and if they are specific for each type of odor, Wilson. John Lopez,
Uh, the protein that bind the others, you mean OBPs?
yes, I think it should be OBPs.
Yes, sorry, but I'm here not study OBPs because OBP is more something general, is not more specific because we are looking for odorant receptor that's more specific for a specific compound. I think OBPs can bind to several compounds. Then I don't know, Eugene, if I'm wrong, please correct me. I don't know how many genes of odorant receptor OBPs are present in insects. I don't know if it's like that much like in like for
Yeah,
ORs.
they do have a lot of genes, Wilson, covering that. They are not so specific like the receptor
Yeah.
themselves, but they do interact more and less with more and less affinity to specific kind of chemical groups. Let's say some of these OBP are more, specialized to bind to cetones groups. Other are
Okay.
more specific to bind to alcohol,
Gotcha.
with short of long,
Hmm.
chains, So they do have some. They are. We used to say, and John know that because I'm more than sure that his insect physiology professor explained it to him. I don't know, 2 years ago maybe, uh, saying that they are the first center for filtering these bunch millions of odorant molecules that are entering the sensilla every time. So
Yeah.
this kind they do a first very raw selection of the groups and bring that to the receptors in the uh, dendrite membrane.
Hmm.
So, and then from there one that they can,
Yeah.
go bring the information to the antennal lobe, and from there send
Yeah.
the for canyon cells.
Exactly.
But
Yes. Is much more general, and yes, the insect should have this filter because
yes,
a lot of information out there. And thank you for complimenting my answer. And yes, you mentioned something very important because that odorant receptor that is expressing in that neuron there in the syncytium, that connect the axon of that neuron connect to the antennal lobe, is the first connection to the brain. I didn't show that because it's not the topic, it's not the central. At the brain level, my talk was just the peripheral. But yes, you mentioned something very important because uh, each each neuron from the syncytium connect to uh, uh, a glomeruli, glomerulus in the antennal lobe.
okay, Wilson. And the other question, Ezra is asking us another question here. Is it possible to have mosquitoes, susceptible mosquitoes to pyrethroids, having knockdown resistant genes?
Yes, yes, yes. For example, this. Yes, you mean knockdown resistant genes is like KDR and KDR genes, KDR mutation. Yes, there are some species of mosquito insect that they have the mutation there, but the mutation is not in reduce the effect of parathyroids. I mean, it's like a silent mutation, I would say, because they have the mutation, but the mutation is not reduce the susceptibility. Okay, I don't know. Maybe the mutations change the property of the channel in another way, but yes, susceptible mosquito can have KDR genes. Yes, it's possible, but most of the time the genes will make the mosquito resistant. Resistant. Okay.
Okay. There is another very good question here from Vanessa Wilson. Is there any evidence of insects being attracted to infected hosts? If there are, which receptors are activated in that case?
You
Those
mean,
cases.
okay, infect the host, you mean like dengue, Zika, I don't know. Okay, no, I never heard about that. I mean, I would say that they infect people not to be more attracted. It's not like I think the pathogens cannot change our smell because the most important attraction, most important stimuli for mosquito is the CO2 and the smell from our body. I think the pathogens will not change that. I think for plants, yes, for plants it's different because the pathogen can change the volatile of plants. But for us, I think no. I would say no in a first moment, but I need to read more about that. Okay.
I would say yes, Wilson. I would disagree with you. If the pathogens can change the smells of plants, why not by us, humans or animals?
I yeah. I mean, yeah, but I never look in that way. I mean, I never check paper like that to see if
Yeah, me neither, but I would expect so.
yeah,
Uh,
maybe because they can change our temperature. For example, yellow fever, you have
yes,
fever, and you are warmer and more
yeah.
warm, and then this
And remember, this I learned from you. I mean, as at the same time we are food, but we are also predator for the mosquitoes. Also, if you are,
exactly. That's the like how sophisticated mosquitoes are to detect a prey and at the same time that prey can be a predator. It's amazing how they can balance these two things, right? I mean, yeah, they are there on skin feeding, they can be killed in time.
okay, John, say thank you, Dr. Wilson and teacher Eugenio. Your works is very amazing. Congratulations. We think of you, John, and all the people that here present today. Was a nice moment, you know. And Douglas has another question. What is the difference between Pyrithione and Pyrithrine? Because I read something of Pyrithrine is banned, Wilson.
Mm. Okay, like pyrethrine extract is the extract from plant. Okay, it's the crude solution. It's a solution with several compounds. Inside that extract with several compounds, you have pyrethrines. Pyrethrines have like you have two types of pyrethrines: pyrethrine one and pyrethrine two, and it's like if you make the composition of that structure, see the component, we have these pyrethrines, okay? Then people purify this pyrethrine and use that structure to make pyrethrines. That's the major difference between pyrethrum extract and pyrethrin. Both target the same target, sodium channel, okay, insects as an insecticide. Uh, why do you mean Pyrethrine is banned? It was not available anymore to buy? Because, I mean, we can buy Pyrethrine, but the problem of Pyrethrine and Pyreto, they are not very stable. When you apply outside, they will degrade very fast. That's why people change the structure to make pyrethroids more stable, not degraded by light, for example.
I maybe Douglas is make Douglas. I don't know. Maybe you are confusing with pyrethroids that are the synthetic compounds. Some pyrethroids are indeed banned for a long time, but pyrethrines I do not expect to be banned because they are not so, I mean, toxic for humans. The problems with pyrethrines are more related to as Wilson said, the stability of these molecules in the environment. So they degrade very fast. So it's not good for any company produce these things to, you know, to sell. So that's why people 50 years ago developed the pyrethroids, trying to solve such problems. And I'm more than sure if you can produce such pyrethrum. Pyrethrum is only the crude extract, like Wilson
Yeah,
said, from the chrysanthemums. You can use it here in the Brazilian agriculture, organic production for sure, because you can use even nowadays, for our big surprise, you can use, tobacco solutions that has tobacco on the tobacco extracts on there, and that you can use still can use it in sell the product, saying that it's organic production, and that tobacco has a lot of nicotine, right? And nicotine is very, very dangerous for humans and all the other animals. That's a big surprise to me, Douglas. But it's okay. Nice. Vanessa has another two questions, I think, and Ezra is another question. Let's see here. Ezra, did I ask you? Did I miss you one a question? Okay. Is it possible to have mosquitoes susceptible mosquito to pyrethroids? This one is already answered. I asked a question above. I think we answered that. Let's see, Vanessa. I say yes too. Some species of Lutzomia are attracted for hosts with leishmania, but I don't have an idea what is the bait or mechanism for that attract. As we said here, Vanessa, we are also not aware. Wilson and I expecting that indeed some of these, it depends on the infection type, but some mosquito or some other insect vector can be more attractive for hosts that are,
Yeah,
ill and stuff like that.
yeah.
But
In general, mosquito can tell apart which prey, which host they like most. Okay, because if you are gathered together outside, very likely there is one single person there that get more bitten than others. Then yes, they can they can they have this preference for one person to another person. It's related to smell. If the a sick person is smelling different, that's more attractant to mosquito. Yes, they will be attracted to.
okay, Wilson, we went by here for the 1 hour and 40 minutes. Thank you very much again for your
Okay.
help, for your time, for your, nice delivery presentation. And I would like to thank all the students, all the people that came to your talk. It
Thank you,
was a
guys.
big pleasure. And for those students that are doing the class this year, it will be a big pleasure to me in the near future having you guys on the other side, like it's happened here today with Wilson. I think that our graduate school need to bring this stuff for we have a lot of people that did master or PhD in our program, then went outside, and I think we can invite these people to contribute for being here with during the course or even during the seminars as well.
Hmm.
Okay. Thank you very much, Wilson. Have a
Thank
nice
you.
weekend there.
You
Enjoy.
too. Yeah, I still need to go to the lab now. I need to collect some RNA there. Oh my goodness. Yeah,
Enjoy.
my happy hour is gonna be on later.
Ok.
Later today.
Sextou com S de saudade, tá, gente?
Yeah, Friday.
Friday.
Friday com F de fui. Bye, guys.
And thanks God this Friday, right? To keep
Yeah.
it in English.
Yeah. Thank
Okay.
goodness. Thank goodness it's Friday.
Okay. Bye, guys. Thank you very much. Thank you, people from Insectum, Wallison, Douglas, Jessica, all of you. Thank you very much.
Thank you guys. Thank you very much for teaching a lot here. Bye bye.